The topics I have chosen in topics in biotechnology is engineered miniature CRISPR-Cas system for mammalian genome regulation and editing. Before going on to the topic, founder thank Jennifer Doudna, is the founder of CRISPR. The DNA sequence family known as CRISPR, also known as cluster regulatory interspace of palindromic repeats, is present in the genomes of prokaryotic organisms like bacteria and archaea. This sequence were from bacteriophage DNA pieces that had previously infected the prokaryote. When the new infection occurs, they are used to find and eradicate DNA from related bacteriophages. Therefore, this sequence play a crucial part in the prokaryotic organisms' antiviral defense mechanism and offer a sort of acquired immunity. Nearly 90% of sequence archaea, about 50% of sequence bacterial genome contain CRISPR. This clinical trials using CRISPR therapeutics to treat individuals with congenital disorder have recently made groundbreaking advancement. Despite this advancement, the physical size of current CRISPR gene editing nucleus restrict their use in clinical setting due to the difficulty in effectively delivering large molecule into target cell, as they do not fit in the cargo space of clinically safe viral delivery vectors, or they impair the transport capability of the nanoparticles. Therefore, the size is a significant barrier to the therapeutic use of gene editing drugs. Overall, 99.9% of known CRISPR nucleases do not function in mammalian cell, which is the main issue we are confronting. To neutralize invasive phases with compact and straightforward genomes, prokaryotic species develop CRISPR defense mechanism. Therefore, even if we add localization signal to the Cas nucleus to solve the issue of getting it into the mammalian cell and across the nuclear membrane, the vast majority of natural CRISPR systems still lack the ability to find and access their DNA targets among strands of millions of nucleotides. Given the slim chances, researchers choose a candidate from class 2 type V5 family of extremely small RNA-guided nucleus, which is Cas12F, and its goal was to determine whether or not molecular engineering might transfer the little Cas12F protein into a useful CRISPR tool for usage in the mammalian cell. So, onto the onto the topic. Cas12F does not function in mammalian cell, and engineered Cas12F mutants named Cas-mini show comparable activities with Cas12A for efficient gene activation. Cas nucleus allow efficient and specific genome editing, while nucleus deactivated decas molecules fused with transcriptional or epigenetic effectors enable targeted regulation of indigenous gene in mammalian cell via programmable binding to DNA. These systems offer opportunities for development of gene therapies against a variety of genetic diseases. Interestingly, all the designs improve gene activation with designed to outperforming the other two. Engineering the Cas12F protein for enhanced gene activation in mammalian cells to test the effect of Cas12F protein on gene expression, we use an iterative protein engineering strategy. The iterative protein engineering and screening yielded a gradually improved Cas12F parent library with D14-3R, D14-7R, K33OR, K528R variants showing the almost 200-fold improvement of reporter gene activation over the wild-type Cas12F, which is from now this variant is termed as a Cas-mini. We next whether the Cas-mini PPR could activate indigenous gene on SBG-IL-1RN and SACL in HCK293T specialized cell. So it shows this single guide RNA, so the gene is highly dependent on single guide RNA targeting site. The efficiency The characterization activation in this cell using the library of single guide RNA for human indigenous genes, interferon gamma, CD2, CXCR4, SBP. 10 single guided RNA were tested for every gene except for the SBP, on which 20 single guide RNA were tested and show the fold of mRNA activation. So Decasmani for PPR outperforms the Decas12F PPR for indigenous gene activation and is comparable to Decas12A, with both effectors using the optimized single guide RNA scaffold. For all single guide RNA tested, we observe consistently and enhanced activation for each gene. For example, interferon gamma activation using two different single guide RNA, D-Cas-mini BPR outperformed 45-fold or 120-fold better than Decas12F PPR on the mRNA level measured by quantitative PCR, or 25-fold or 7-fold better than Decas PPR on the protein level measured by ELISA. Comparison of Cas-mini to Cas12F and Cas12 for gene activation. The experiment were used a single guide RNA designed to undoing the experiment, the three indigenous genes and designed Cas12a crRNA binding to the same target sequence of the best performing single guide RNA of Cas-mini, so was outperformed by the twofold. For the most tested indigenous gene, we observed that Cas-mini outperforms the Decas12a BPR. Uh, PPR for gene activation. The nucleus active Cas-mini enables robust gene editing in mammalian cell. So we next test whether the nucleus active version of the Cas-mini variant could cut and edit genome DNA in the human cell. We observe robust gene editing in the Cas-mini, which bring out to the conclusion that Cas-mini variants enabling optimal gene editing can be different from those of those used for base gene activation. Consistently, the results using the Cas-mini show that in vivo gene editing also peaked around position 22-30 base pair relative to the palm. We thus confirm that Cas-mini can be used in broad genome editing application in addition to gene activation. The efficiency of Cas-mini effector is comparable to Decas12a system when tested for gene activation or base editing. Our engineered Cas-mini variants show significantly improved gene activation over the wild-type Cas12F, suggesting that enhancing the interaction in the Cas12F single guide RNA and the DNA complex is important for the both application. Cas-mini may have different gene editing or gene regulation features compared to Cas9 or Cas12a. The principle for designing efficient single guide RNA at different chromatin regions for gene activation or gene editing remain unknown, but needs to be clarified. It in addition to the gene editing, base editing, and gene application demonstrated in the study, Cas-mini can be potentially used for the genome engineering application, including epigenome editing and chromatin engineering. Thank you.